to talk a little bit about diffusion. And diffusion, as I have it defined here, is the passive movement of solute in solution or across a barrier. So as you'll see, we'll have to actually explain diffusion under two different circumstances. The reason to talk about diffusion is that it is an enormous factor in the functioning of many physiological systems. And what we mean by that is that in many cases, diffusion is what is allowing solutes to move from one location to another. So for instance, as our cells produce carbon dioxide, it diffuses out of those cells and into the blood. The same is true when we get to the lungs, the opposite direction. But when we get to the lungs, carbon dioxide would diffuse out of the blood and into the air so we can exhale it. Oxygen moving just the exact opposite direction. And so diffusion plays an enormous role in general physiology at the level of the tissues and organs all the time. The other main reason to talk about diffusion is that it is an enormously limiting factor and is therefore one of the evolutionary driving forces for the evolution, the development of many of our organ systems. In other words, why have this system of heart and blood vessels that move blood throughout the body so rapidly to move oxygen and carbon dioxide? Well, the rationale behind that, the adaptive purpose, is that diffusion, if we were to rely on diffusion to get oxygen from our cells all the way to the exterior of our bodies for waste removal, or again, the opposite direction, to get oxygen into our bodies and all the way to our cells, we'd be talking about an exceptionally slow process, right? So we've evolved many of these systems to sort of overcome the limitations of diffusion. Okay, so what is diffusion again? So it's the passive movement of solute in solution or across a barrier. So we can come up with sort of a very simple example here, one of which you've probably even seen before. And that is sort of thinking about diffusion as the dispersal of, let's say, a dye in a cup. Right? So we've got some water in here. Right? And if we were to put a drop of dye into this cup, this is sort of time one. And actually, let me back up. We would actually start with, if we show the dye as individual molecules, right? So we have our cup here. It's got fluid in it, and we add our dye, and the dye, of course, is individual molecules that we can't see, right? But this dye would be very high concentration right where we add it to the solution, right? And so that's at time 0, right? T equals 0. And then over time, and this is without any sort of stirring or anything else, what's going to happen, right, is this dye will end up sort of equally dispersed throughout this solution. Now, it may take a while, depending upon the amount of time and some other factors that we will talk about in a moment, but the dye is essentially going to disperse outward from this sort of high concentration region in all directions, three-dimensionally, right? And so diffusion is actually powered by random molecular motion. Molecules at anything above what's called absolute zero, at which a really low temperature, minus 273 degrees, anything above that, molecules are in motion, right? At absolute zero, even molecules stop moving, which is a crazy low temperature, right? But anything above that, molecules are moving just because of the thermal energy, right? And so they're bouncing into each other. And so on the left here, the molecules are very close together and they're bumping into each other and then being repelled outward by those collisions. And then the farther they get apart, they might still be bouncing in each other. They're still diffusing, but there's no longer what we would call net diffusion, because the molecules on this side are diffusing this direction, and the molecules on this side are diffusing this direction, and the top to the bottom, and the bottom to the top. And so the net overall effect is zero, right? Because they've already equally dispersed within the solution. Whereas on the left, we would have net diffusion, right? Because we're going to go from a high concentration here, right, at this spot, to everywhere else in the solution, which is at a lower concentration. Actually, concentration is zero initially, right? And so we could have, time over here is just simply greater than zero, whatever that time may require, right? And so let's talk about, there's an equation to explain the factors that affect the rate of diffusion. And so we're going to use what's called Fick's law. And it's actually Fick's first law. He has multiple laws of diffusion. But we're going to stick with a pretty simple one. And what it says is that J, which is flux, and then I'm going to add, there's lots of types of flux. So we're going to add a little key down here to the bottom. So J diff, so diffusive flux, so diff for diffusion. So diffusive flux equals negative dA times delta C over delta X. I'm going to explain all of these in just a second. Okay. Now, so what are all these elements? Well, let's do some of the easier ones first, right? So remember, J diff is the rate of diffusion. And so that's going to depend on, let's start with D here. D is the diffusion coefficient. Oops. The diffusion coefficient. And that has a multitude of things sort of blended into it to account for a variety of things. And those include the size of the molecule, right? And, oh, by the way, so diffusion is directly proportional to D, A, and delta C, because those are all on the top, right? In other words, there's a fraction over here, so the delta X is on the bottom. But what that means is that if D goes up, so does J, right? Or if A goes up, so does J. That's called directly proportional. It's really important because there are a lot of fractional relationships in physiology where there's, a numerator, right? D, A, and delta C, and then there's a denominator over here, delta X, right? And so everything on top is directly proportional. In other words, they go up 1 to 1, right? If A is doubled, J diff would be doubled. Right? And so again, all the components of D would be directly proportional as well, because they're all within D and it's on the top of the equation over here. Okay, so let's get back to this. So size. Size is a factor because the larger the molecule, the more what's called molecular drag it has. It's just harder to move a big molecule through a solution than a little tiny one. It's bumping up against more water molecules the larger it gets. So that's pretty literal. Makes pretty good sense, I think. Then there is viscosity. And viscosity is a characteristic of the solution, not the solute, not the molecule that's moving through solution. And remember this idea of a solute. We could be talking about sodium or chloride, any ion. We could be talking about a nutrient, a waste, any of these things, even proteins, right, are solutes. But the solution they're moving through comes into play because of its viscosity. And the more viscous it becomes, the more thick, if you will, the solution becomes, the harder it is for things to diffuse. And again, if there's a fractional relationship within this. And so if viscosity goes up, then J diff goes down, and we're not showing you that within this. But just let's leave it as viscosity is a component of diffusion, and then let's just use logic. The more viscous a solution is, the slower the diffusion would become, right? Okay, and then if we come down with one more, we've got temperature. Right, and we said diffusion is driven by the passive movement of solutes. And that's generated, that's powered by random molecular motion. Well, guess what? Random molecular motion increases when you increase temperature. Molecules move faster when they're hotter, right? When they have greater thermal energy. Okay, so that's the diffusion coefficient. All right. Then we have A, which is the surface area. And this is the surface area across which the diffusion can occur, right? It's a little hard to sort of visualize when we've got this sort of open glass of water here and a dye. But the drop of dye is in contact with a particular surface, right? With a certain amount of fresh water around it, non-dye containing water. It'll be a little easier to understand when we have a membrane, right? We'll talk about that in a minute, right? Because we said up here at the top that we could also have diffusion across a membrane, in which case that membrane has a very much easier to comprehend surface area, right? Because it's a physical structure. Okay, but again, directly proportional. So if the surface area goes up, the rate of diffusion will go up. This is easily demonstrated by the lungs, where we have a very large surface area due to structures that are called alveoli. They're like little air sacs. But by having hundreds of millions of those, we increase the surface area dramatically, which enhances our diffusion across the lung surface. All right, bumping over here to delta C, we have, so that's the concentration gradient. So the larger the concentration gradient, the bigger the difference between, right, if this was sort of a dilute dye, the difference between the dye area and the water area, I'm pointing up here at the top now, right, in this picture, right? If this was a very concentrated dye, it would be, the gradient would be larger between the dye and the fresh water, and we would have faster diffusion, right? Because the concentration gradient would be larger, the difference between the dye and the non-dye areas, right? And so I like to say that the diffusion, this net diffusion, as I defined it before, remember diffusion is occurring over here, but it's really not changing anything because it's already mixed. Whereas net diffusion, right, before it's mixed, occurs because of a concentration gradient. Right? So that's important. Concentration gradients drive net diffusion. Okay, and again, the bigger the concentration gradient, the faster the diffusion. Again, we'll come to that when we talk about capillary exchange or how the lungs work and having a large enough gradient to drive rapid diffusion. It's really important. All right, the last factor over here, right, is that delta X. Again, delta means the difference in, right? So this is the difference in distance. And how that comes into play is that the farther we need, let's say if we made our cup larger and we put the same amount of dye in it, right? Because it would be diffusing a greater distance to all the way over to a farther corner of this larger container, right? It would take longer. Aha, now we are at inversely proportional. In other words, as distance goes up, right, as this increases, this is on the bottom of the equation. So as this increases, our diffusion over here would go down, right? Everything else was directly proportional. Those, when D, A, or delta C went up, diffusion went up. But if delta X goes up, the distance goes up, then diffusion goes down. That's called inversely proportional, right? When they go in opposite directions, not in the same direction. Directly proportional, same direction. Inversely proportional, opposite directions. Okay, that is the basics of Fick's first law of diffusion, describing movement of a solute in solution. Now we have to add one more complexity here, and that is we're not always diffusing through water, right? Sometimes when in biological systems, substances have to diffuse across membranes. And so the issue there becomes, we need to know if this substance is soluble in a membrane. Right, membranes are made what are called lipid bilayers, right? So it has to have a lipid solubility. So we have to add that component to this equation, right? And I can actually even draw that over here, right? If we were to have our cup of water again, right? And now we were to add a membrane. Let's make it green. Right, down the middle of this, I'm going to give it some thickness, and I have it sort of dashed here because it has to have it has to be permeable if we're going to have any diffusion at all. Right, now let's add our dye again. Adding our dye, but now we're adding our dye on one side, right? And it would diffuse within this one side, but it would also, right, over time, it's going to diffuse. It's going to diffuse over here, right? And it's going to end up sort of equally dispersed here. But as it's doing that, it also has the potential to diffuse across here. But it's like, well, does it or doesn't it? And so let's give you the new, or slightly modified, I should say, equation for diffusion across a lipid bilayer. And it's still J-diff. It's just diffusion in across a different medium. Instead of through water, right? Now it's going to be diffusion across a membrane. And in that scenario, it's still negative dA, but now we have to add this factor. Let me give it a color here. We have to add a factor, which is lambda. And then still times delta C over delta X. Everything else is the same, but we've got this guy, right? And that is our lipid solubility factor. Right? And so if the solute that we're talking about here, this red solute, has a lipid solubility of 0, right? We have a question mark here. Would it be able to cross this? Well, if lipid solubility is 0, right? In other words, if that were 0, what does J-diff equal, right? Well, you're multiplying everything by 0 over here. So of course there would be no diffusion across this barrier, this membrane, into this second compartment, right? However, in biology, there are substances that have fairly significant lipid solubilities. The gases, for instance, oxygen and carbon dioxide have relatively high lipid solubilities. And so if that's a very high value, then as long as it's not limited by other factors, then diffusion can be quite good, as it is in the lung, or as it is into and out of capillaries, exchanging at the tissues, right? And so in this scenario, one other thing to point out is this delta X. What distance are we talking about here? Well, that delta X is literally just the thickness of the membrane that we're diffusing across, right? Because we're talking about the solubility across the barrier, the diffusion across the barrier. Once you're over here, right, and you're talking about getting from here to here, we no longer need the lambda. So, right? So really the problem with diffusion is that the factors that describe it differ slightly depending upon whether you're talking about in just a general solution like this, or if you're talking about crossing this lipid barrier like that, right? And so again, you don't even need to use the equation for this course, but what you do need to know is the factors that affect diffusion and how they affect diffusion, i.e., are they directly proportional or is it inversely proportional, things like that. And also why diffusion is so important, right? It's important for exchange between compartments within our body, from the cells to the interstitial fluid, from the interstitial fluid to the blood or plasma, and then the same thing at the lungs, right? We can talk about it in multiple locations. But then finally, the driving force of diffusion for organ system evolution, right? The fact that diffusion is so limiting over distance. In fact, if we talk about the time that diffusion takes, time for diffusion, it's proportional to, that's this symbol right here, that's proportional to delta x, the distance, squared, right? And so instead of it being, right, some time changing directly with delta x, it's still directly proportional, but we've got this exponent, right? Which means as delta x, as distance increases, the time for diffusion goes up exponentially, right? So if it takes, let's say it takes a couple of milliseconds for a solute to diffuse across a cell that's 10 microns across, a millionth of a meter, right? So diffusion across this little cell, let's say that takes a couple of milliseconds. Well, let's now have a cell that's 10 times bigger, right? That's about 10 times bigger, right? So if this was a couple of milliseconds to diffuse across that cell, what's it take to diffuse across? And I mean literally across from one side to the other, right? Well, it's not 10 times. Remember, this cell, this bigger cell is 10 times larger than the smaller one. It's not 10 times longer. It's 100 times longer, right? Because of the exponent here, right? This is hugely important. And what it means is that the bigger things get, or the farther our diffusion has to occur across, the worse it gets, right? The longer it takes, in which case it might not be effective. It's why capillaries need to be really close to cells, right? You can't have a cell here and a capillary over here, right? And expect to get oxygen to diffuse all the way to this cell in any reasonable amount of time. We have to have lots of capillaries. So there's a capillary here, and there's a capillary on this side, right? And we get good diffusion because our delta x, right, this black line, is really small, right? The smaller x is, the shorter the time will be for diffusion dramatically. Okay.